Hello and good day. I am Salubun Michelle D and this is the performance of the Group 4 for the finals return demonstration on MLS 401 laboratory. In this video, we are going to perform infiltration, embedding, floating and fishing out, H&E staining, and mounting. So after the tissue has undergone the process of fixation, decalcification, dehydration, and clearing, it will then proceed to infiltration. So, infiltration is the process wherein the tissue is placed in a paraffin wax bath or any medium of choice. This is to make sure the tissue is stable and homogeneous, which makes it suitable for sectioning.
For the process of infiltration, I used the following materials: a frying pan, aluminum foil, petri dish, tweezers, paper towel, candle, chopstick, thread, gauze, and little jars. Prep the specimen on the decalcification setup. Use chopstick, thread, tweezers, gauze, and the specimen. Make sure that the specimen is labeled and the label is placed where the specimen is and not on the cover. This is to avoid cross-examination. Put the other materials aside but nearby to make more area as you perform this procedure. Get the tweezers to pick the specimen and transfer it into the gauze. Fold the gauze and get a piece of thread and tie the gauze with the thread. After the gauze is tied, get a chopstick and tie the ends of the thread into the chopstick. To avoid losing the specimen on the process, put a label on the chopstick where the specimen is tied to. Put the prep specimen aside and prepare the paraffin wax. Get the frying pan and cover it with aluminum foil. First, preheat the frying pan. Then put the candle in the frying pan and wait for it to melt. As the candle melts, the wicks are visible and are accessible to be picked, so gather the wicks from the melted candle. Next is the paraffin wax bath. Get the container from before and open it. This is where the tissue will be immersed in the paraffin wax. Get the melted paraffin wax from before. Since it is not easy to transfer the melted paraffin wax from the frying pan into the small container, I used a measuring cup. The specimen will undergo 3 changes of paraffin wax bath with its corresponding intervals. And this is the first paraffin wax bath. Get the specimen and make sure it is the right specimen by looking at the label. Put it into the paraffin. The first paraffin wax bath recommends a 30 minutes interval after the paraffin wax is changed to the fresh ones. So leave the specimen in the paraffin wax bath for about 30 minutes. Make sure that the temperature does not exceed 60 degrees Celsius to not create a substantial tissue damage. After the 15 minutes is done, get another jar or another container. Fill the jar or container with a fresh set of paraffin wax. Then transfer the specimen from the first paraffin wax bath into the second. Leave the specimen into the second paraffin wax bath for another 30 minutes. After 30 minutes, at last it is the final paraffin wax bath. Same as before, get a new container and fill it with another fresh batch of paraffin wax. Transfer the specimen into the third paraffin wax. And since it is the last change, leave the specimen in the paraffin wax bath for about 45 minutes. After the 45 minutes is done, get the tissue, remove it from the wax, and place them on the paper towel. The sample is then ready and we'll proceed to embedding.
Now we have here our infiltrated specimen with its accessioning number SP21666. Now after infiltration, the next step in tissue processing is called embedding process. It will be demonstrated by yours truly, Julian May D. Manjala. The embedding process is where the tissue specimen is surrounded by its infiltrating medium. After such, it serves to be a sufficient external support during sectioning, and it could also be beneficial for archival purposes as the pathologist can easily retrieve, recut, and re-examine the tissue block for further examination and analysis. Now let's move on to embedding the tissue specimen. We have here our infiltrated tissue specimen, our embedding medium, which is the melted paraffin wax, and lastly our embedding mold, which is a disposable paper mold or a paper boat, which can vary in sizes from small, medium, or large.
First step is to take out the tissue specimen from the cassette and assess whether it should be placed in a small, medium, or large embedding mold. Next is prepare the desired mold and pour in a bit of a melted paraffin wax to act as a base. Place the tissue specimen in the middle of the mold and flatten it out. Let the tissue specimen sit for a few minutes for it to be secured in the melted paraffin wax. After, dispense a decent amount of melted paraffin wax onto the mold in order to completely form the tissue block. Let it sit again for a few minutes and wait until the tissue specimen will be a firm tissue block. Since I've said earlier that it can be served for archival purposes, the tissue specimen can be stored for up to 10 years. Thus, labeling the specimen is a must. And for this specimen, we have the accessioning number SP21666.
If it can be easily removed from its mold. Now, let's go in to check it. So here is our tissue specimen. Now this tissue specimen is now ready for sectioning.
The paraffin section flotation bath is designed with handling of paraffin wax samples in the histopathology laboratory. The water bath is an essential step in histopathology processing because it allows our ribbons to flatten out and stick to the glass slide when placed. The materials that we are going to use today are egg white, our glass slide, and here is our flotation bath.
Hello everyone, I am Andrea Kong and I am tasked to perform the H&E staining method. So a little overview about what I am about to conduct. H&E staining, also called as hematoxylin and eosin staining, is a common laboratory method that uses two dyes called hematoxylin and eosin. It enables to see morphological changes of the cell under a microscope. It also helps identify different types of cells and tissues and provides important information about the pattern, shape, and the structure of cells in a tissue sample. Here are the materials that we will need. In the first part of the experiment, let us conduct the fishing out method of our tissue ribbons from the water bath. Do not forget to label the slides. Then, prepare the slides containing the paraffin section and allow the slides to cool down for at least 5 minutes after being placed in the slide warmer. So let us proceed to the procedure proper. Start the first method of staining by immersing the slide in saline. It must be changed 2 times and should be dipped for 5 minutes in every change. Next is dip the slide in alcohol formol 10 times. After that, use 3 changes of 95% ethanol, then dip the slide 10 times in every change. Then using soapy water, wash the slides and use a cotton swab and carefully remove the excess albumin or impurities. To clear the slides from soap, wash thoroughly with clean water, Then dry the slide using a paper towel. Make sure not to touch the tissue specimen.
Stain the slide in hematoxylin for 5 to 10 minutes. After, drain excess stain, then rinse with water. Decolorize sections in acid alcohol by quick dipping it 2 times. Rinse slides immediately in water. Immerse slides in ammonia water for 5 minutes or until it turns blue. After, rinse again in water. Drain excess water on paper towels. Stain slides with AUSIN Y for 1.5 to 3 minutes. Drain excess stain, then rinse well with water. Drain excess water on paper towels. Dip in 95% ethanol for 3 changes with 10 dips each. Lastly, dip slides in xylene for 3 changes with 10 dips each. Wipe the edges and the back of the slide to clean it. Take extra care not to touch the sections.
Hi, I am Mariah Carla M. Cabaisin, and I'm gonna be performing mounting. Add 1 drop of mountant into our cover slip. Then, let's mix it with xylene. Then, gently push it into our slide. And let's wipe off the excess medium. That's it. The purpose of mounting is to preserve the slides for storage purposes and to prevent abrasion or staining.